Okay, so now the front line almost reached the bottom of the gel. So we are about to stop the gel running, and now we can start to prepare the transfer buffer. Okay, so how to prepare? We add 700 milliliter of the DI water and 100 milliliter of the 10 times concentrated transfer buffer and 200 milliliter of the methanol. Okay, so now I'm going to add 100 milliliter of the 10 times transfer buffer to the cylinder. Next, we are going to add 200 milliliter of the methanol. Good enough. Okay, now I'm going to pour some into the transfer container. Okay, we are going to set up the transfer system in this container. Okay, so basically this container has two sides. Okay, and this is the transfer cassette. Okay, it has the black side, red side. Okay, and this is the foam. This is the paper, which will be put around the gel and the PVDF membrane, okay, to support the transfer. Okay, and this we are going to use the wet transfer method. So what I'm going to do, I wanted to wet the whole system, okay, by pouring, the transfer buffer to completely wet the system. Okay. And also, I'm going to pour some in this side. Okay. And this is a tool later we are going to use in order to get rid of the bubbles. Okay. So currently, I'm going to leave it here. Okay, so basically now I'm preparing the transfer. Okay. Okay, what I'm going to do now, I wanted to take out the gel. Okay. Stop the running. Okay, and I'm going to okay, so I'm going to take out the gel. For the remaining running buffer inside the cathode side, I can pour it here. Okay. So now the gel is in between the two plates. Okay. So I'm going to separate them. Okay, with this tool. Okay, I'm going to insert in between. Okay, here there is the two plates. Okay. Okay, I'm removing this plate, and now the gel attached to this plate. Okay. So what I'm going to do, I'm going to cut the additional gel without our samples. Okay, so I'm going to cut from the right side of the last pre-stain marker. Okay. Okay, I'm going to get rid of the right side of the gel. Okay. Okay, and I'm going to also cut off the top part of the gel, okay, which is the stacking part of the gel, okay. Okay, I think I'm going to cut from here. Okay, you don't want to cut between your protein area, okay, separating gel. I wanted to just cut from the top. Okay, next, I also needed to make one more cut, okay, because we loaded the sample in triplicate, okay. So next I'm going to cut, let's do it in the middle of the first ladder, okay. And I'm going to use the left side of the gel for the Coomassie staining and the right side of the gel for the Western transfer, okay. So I'm going to cut in the middle. The reason to cut in the middle, I don't want to damage, your protein loading length, okay. So I'm going to cut in the middle. Okay, and I'm going to put the left side of the gel into this container for the Coomassie staining, okay. So when you grab the gel, do it gently. Okay, I'm going to put it here, and then pour the Coomassie staining dye buffer, okay. Now we are going to stain the because now you cannot see your protein bands, okay. It's transparent and very clear, okay. In order to see the protein bands, we wanted to pour the Coomassie staining buffer, okay. And we are going to stain the gel for maybe 1 hour. Just gentle shaking is good enough. So what we are going to do next, I wanted to put the gel, okay, into the transfer buffer, okay. Okay. Okay, I wanted to completely immerse the gel into the transfer buffer, okay. Okay, next we are going to activate the PVDF membrane, okay. So this is the PVDF membrane we are going to use, okay. And we already cut into the right side we needed to use, okay. Okay, and the middle part, the white colored one, is the PVDF membrane, okay. But in order to protect it, there are two papers, surrounding, okay, on the front and the back to protect it, okay. So what we are going to do, I'm going to add some methanol into this container. Okay, and I'm going to put this inside. Okay, when you handle the PVDF membrane, okay, you don't want it to fold. Okay, you want it to grab the corner of the PVDF membrane and don't use your finger to touch it. Okay, because we are going to transfer the protein from the gel onto the surface of the PVDF membrane. Okay, you don't want it to cause some contamination, okay, or make it dirty. Okay, so I'm going to grab one corner, okay, and then I'm going to immerse PVDF membrane into the methanol. Okay, the reason of this is we wanted to activate, the surface binding ability of the PVDF membrane. We are going to keep it for 1 minute. This is our transfer apparatus, okay, which is different from the SDS PAGE gel running system. Okay, there is a black side and red side. Okay, and I'm going to take out the PVDF membrane and immerse also into the transfer buffer. Okay, so also grab the corner, okay, and immerse inside. Okay, okay. Okay, and then we are going to set up the transfer system. Okay, how to set up the transfer system? Basically, we need to, it's a sandwich system. Okay, so what we are going to do, this is the black side of the cassette. Okay, we put a foam, okay, on the black side, on the top of the black side of the cassette. Okay, and what we are going to do next, we wanted to put a paper, a supporting paper. Okay, this is relatively thick. Okay, in the middle, okay, of the black of the foam. Okay, and we are going to, because the protein will be transferred from the black side to the red side. Okay, so next we are going to put the gel onto the paper. Okay, so when you handle the gel, handle gently. Okay, we wanted to gently lay the gel on the supporting paper. Okay, so next we are going to lay the PVDF membrane, okay, onto the gel. Okay, and remember, do not introduce bubbles. Okay. Okay, and in this way, the protein will be able to move from the gel to the PVDF membrane. Okay, and what we are going to do next, put another paper. Okay, and another foam. Okay, so after we put the paper, we wanted to remove any remaining bubbles. Okay, we don't know whether there are some tiny bubbles inside. So what we are going to do is we wanted to roll. We wanted to roll, okay, gently to try to remove the bubble. Okay. Okay, because the bubble will block the protein transfer to the PVDF membrane. Okay, and then we are going to lay another foam, okay, and close the cassette. Okay. Okay, and you wanted to keep it here, okay, and we are going to insert cassette into the transfer container. Okay, you wanted to put black to black, red to red, okay, so we can do this way, black to black, okay, and this side should face up. Okay, insert it to the bottom, and then we can pour the transfer buffer, okay, into the container. Okay, and we need more. Okay, so what we are going to do, we pour this also into the container, okay. Okay, this is basically some iced blocks. Okay, we are going to, okay, let's see the tip. We can put this one. Because transfer will generate a lot of heat. Okay, so we don't want too much heat. Okay, so basically you want to completely cover, you know, the top part of the cassette. Okay, and what we are going to do, we are going to put the cover, black side and a red side. And this one inside. Okay, so because it will generate a lot of heat, so we are going to put into the ice box, and we are going to put this one into the ice, okay, in order to cool down, okay, during the transfer. Okay, basically you want to use ice to surround the transfer system. Because a lot of heat will be generated, okay? And then I'm going to put on the bench and connect the power supply. Black, black, red, red, okay? And now we are going to turn it on. And we will do the transfer at 100 watts for 90 minutes, okay? 100, okay, 120 is also fine, okay? Okay, let's adjust it to 100. Okay, and the time is around, let's set 2 hours, okay? Okay, so we are done with the transfer. So now we are going to disassemble, you know, the cassette. Okay, so after transfer, we should be able to see the pre-stained marker, the colored marker go from the gel to the PVDF membrane. Okay. Okay, here you can see, okay, clear colored, marker in the membrane, and nothing left on the gel, which means we have a successful transfer. Okay, and what we are going to do next, I wanted to cut off, okay, because the protein will be only on one side of the membrane. Okay, so now it's on this side of the membrane. Okay, so we wanted to mark it. How to mark it? I'm going to turn it over, and then I'm going to cut a corner. Okay, I'm going to cut the right up corner, okay, to mark the positive protein side. Okay, you can grab the non-protein binding area. Okay, and also, okay, we have some extra membrane. I'm going to cut it away. Okay, we still have 2 sets of the samples remaining on the membrane. Okay, and I'm going to cut from the left side of the, or from the middle of the second. Here is the second of the pre-standard marker. Okay, and we are going to use one one part to blot with the anti-EGFP antibody to detect the EGFP expression, and we are going to use the other to detect the beta actin expression. Okay, so I'm going to cut here from the middle of the pre-standard marker. Okay, and I'm also going to cut the right up corner to mark positive protein side. Okay, so remember the right side, right up corner will be our positive protein side. And next I'm going to block the membrane. Okay, the reason to block the membrane is to occupy the non-protein occupying area in the PVDF membrane, okay, to reduce the non-specific binding of the antibody in the following steps. Okay, also try to grab the edge, the corner of the PVDF membrane. Don't grab the middle. Okay, in one we are going to use it. Put it in this container, and the other put it in the other container. Okay. Okay, so we are going to block the membrane at 4 degrees Celsius overnight. Okay, which will be sufficient to occupy non-protein binding area. Okay.